Okay, I'm starting where we left off before we took a break to work some of those amino acid sequence problems. Just want to remind you that this is a depiction of a mass spectrometer. And this is what the data looks like that comes from this. Remember, we were told that the spec measures the mass to charge ratio, and those are reflected by these peaks. The y-axis is the relative abundance. So you see this peak at 678 has very low abundance compared to this peak at 893. So what does this do for us? How do we analyze this? Well, there are a number of bioinformatic databases that have composed the data and mass to charge ratio for a number of, or any variable number of peptide fragments. So what you do is submit your data or your mass spec data to one of these databases, and it searches for peptide fragments that have the same mass to charge ratio. So you can get sequence of these peak fragments, and then based on those, you can overlap them as we did before in some of those exercises we did. So this the protein data bank, the one that's listed here first, I'm probably going to have you search that for a protein at some point in the future, just so you get a look at the information that's provided there. It has about 50,000 molecular structures or fragments for proteins, nucleic acids, and carbohydrates for that matter. So it's just not protein data that is found there. But these are great. Here's a molecular graphic, so you can take your fragments and it can predict what kind of shape might be made by that fragment. So these are great resources for learning things about peptide sequences. Okay, now we're going to talk about protein techniques, of which there are quite a few. These basically are used for isolating proteins and separating a protein of interest that you want to study. For example, we'll look at dialysis, filtration, ion exchange chromatography, salting out, gel filtration chromatography, electrophoresis, affinity chromatography, SDS-Page, HPLC, this is high pressure liquid chromatography, and isoelectric focusing. So let's look at dialysis first. You've probably been exposed to this at some point in a biology class, I would certainly hope. But what we have is a solution of biological molecules that are placed in a semi-permeable membrane. And this membrane has different, well, you can get the membrane so it has different size pores, and we choose our pore range based on the size of our protein. So we can exclude proteins that we aren't interested in. So sometimes we're going to find our protein out here. If our protein is small, our first step in dialysis might be using dialysis or a membrane with small pores, so all our big proteins stay in the bag, and then we'll have to reduce, diacylate and isolate the small protein. So just an example of how that can be used. Ion exchange chromatography also can be used to separate molecules on the basis of charge. So this doesn't have an actual picture of a setup for this, but how this works is We have what are called resins, and these are going to be little beads. For example, this is a polystyrene resin, and it is acidic. So this is used in cation exchange. So we've got a negative charge here. This one is weakly acidic, sorry about that, form of cellulose, and this also is weakly acidic, also a polystyrene chelating agent. So what happens is these resins are poured into a column, so it's a large glass tube. They vary in diameter. So you pour that into a column, pour a buffer over, get it packed nicely, and then you pour your protein solution over it. And obviously you have to know something about your protein based on charge, for example. So is going to be attracted to, a positively charged protein is going to be attracted to this negative, and it will bind the resin. You can wash everything else off and then isolate your protein. Whoops, sorry about that. Here's an anion exchange chromatography resins. These are both basic. This is weakly basic. So we have positive charges here. So obviously we're going to be attracting a negatively charged protein. So here's a better example of, well, here's a little example of what that exchange looks like. So here we have our column that contains our resin. We put a sample in that contains lots of different amino acids. And again, based on the charge, we're going to get a separation of, and here's an example, or trying to depict that, a separation of those polypeptides as they attract to or not attract to the column as you wash those solutions through. This is a collection setup, so these are just little test tubes. So once you get everything washed through, you're going to start eluting the protein, or the fragments from the column. Okay. And that can be done in a number of different ways. Obviously, you're going to add something that's going to compete with binding sites on that resin and get them to shake loose. So let's say we're interested in this particular fraction. You see that the pinks come off, the purples come off, and you do monitor this. There's an apparatus that you can connect this system to that will indicate which test tubes are going to have the elutions. So for example, eluting cations from a column, you're going to use either high salt or high pH, and for the anion exchange elutions, you're going to use high salt or a low pH. So those anionic groups become protonated. So here's an example of that salt elution. You start with a low salt elution buffer and work up to a high salt elution buffer. Again, you're monitoring your fractions coming off so you can identify which tube contains the fraction of interest. Gel filtration chromatography. This separates molecules based on size. So depending, the resin that's used here, these are all some kind of gel beads or resins. They have pores in them. So based on the size of the pores and the size of the molecules, you're either going to get a fast run through or a slow run through. So the bigger molecules aren't going to be able to get into those pores, so they're going to come out first, as you can see here. So here's the elution profile. We get those large macromolecules coming out first, and then the little small molecules, they get hung up wandering around through those pores in those beads. So they're slower to elute, and here's that elution profile here. So depending upon what size you're looking for, you might be interested in this solution or this solution, essentially. There's another separation technique. It's two-dimensional gel electrophoresis. So imagine, or I hope that some of you have been exposed to working with gels. The gel used here is sodium didecyl sulfate gel, and basically we just apply a charge. So two forms of separation here. We're separating based on charge and size. Okay, so if you and the charge is going to be affected by pH. We've got low pH, high pH. So we've got this is the isoelectric focusing part of the gel, and then we have separation by weight in this direction. So these little dots here, and actually obviously this is not a true gel, I'm going to show you one momentarily, but low molecular weight are going to travel faster through this gel, and higher molecular weight proteins are going to move slower through that gel. So that's how we separate them on size going down and on charge going left to right here. This is what a real 2D gel looks like. Not that nice figure that we just saw. But what then can we do with this? We can cut out one of these spots and we can dissolve the gel and analyze the protein that is in that position. Affinity chromatography. So in this process, we're actually attracting proteins either using a metabolite, something it reacts with in the body, or it might be an ion, but something that it binds to. So we can actually isolate specific proteins using its target or its ligand, the thing that it binds to in a cell. So we link that metabolite to our matrix that is in our column. So this, again, it's an insoluble matrix, whether it's cellulose or polyacrylamide in that column. And then we run our protein over it. Our protein that is specific for that ligand is going to bind the matrix or the gel, and then we will use an excess of free metabolite that will displace the protein of interest, and then we can elute that and then use it for further analysis. So we've isolated that protein. So all of these methods are ways that we can separate a protein of interest from a cell lysate. So for example, we have a particular kind of cell, we've broken it open, we've isolated the protein, and now we want to isolate our protein of interest. So there's several ways we can do that, and the protein itself may dictate which method we choose. What I want you to know are not the specifics or the specifics of an elution, but knowing what properties are used in each of these techniques to isolate the protein of interest. So affinity chromatography here was having that target protein, so its ligand. Size and charge in the 2D gel electrophoresis. Those are the types of things I want you to remember and be able to tell me on an exam.